All right, welcome. Today, we're going to talk about winding back the clock, or maybe more accurately, how to read the clock. We are diving deep into the incredible new science that's decoding the very process of aging itself. Okay, let's get right into it.
So let me ask you something right off the bat. How old are you? Now, before you give me a number, just know that it might only be half the story. Because see, you don't just have one age, you actually have two.
So on one side, you've got your chronological age. That's the easy one, right? The number of candles on your birthday cake. But then there's your biological age, and that's a measure of the actual wear and tear on your body's cells. And spoiler alert, scientists are finding that's the age that really counts.
So let's dive into this idea, this concept of your two ages, and why your calendar age really doesn't tell the whole story. I mean, think about it. We all know people who are, say, 60, but seem as fit and energetic as a 40-year-old. And we've seen the reverse, too. Well, that's your biological age right there in action. It's a way better predictor of your risk for things like heart disease or dementia. So the big takeaway is it's not just about how many years you've been around, but how well your cells have held up during that time.
Now, to really understand how we measure any of this, we've got to take a quick trip back in time to a groundbreaking discovery that proved our cells have a kind of built-in expiration date. For the first half of the 20th century, the dominant scientific belief was that our cells were basically immortal. This idea was championed by a Nobel laureate named Alexis Carrel. But then, in 1961, a scientist named Leonard Hayflick noticed something weird in his lab. His cultures of human cells kept dying out, but always after about the same number of divisions. He completely shattered that old myth and started a whole new way of thinking. And this phenomenon got a name: the Hayflick limit. It's this really fundamental idea that our normal, healthy cells, well, they aren't designed to go on dividing forever. They have a finite, pretty much pre-programmed number of divisions before they just hit a wall and stop. So what's the magic number? How many divisions are we talking about? Well, for a typical human cell, it's somewhere in the ballpark of 40 to 60. After that, the cell enters this kind of retirement state. But what's actually going on here? What is counting down these divisions?
Well, the answer is these tiny little structures called telomeres. And the best analogy, the one everyone uses because it's just perfect, is to think of your chromosomes as shoelaces. The telomeres are those little plastic tips on the ends. They protect all that important genetic information inside. And every single time a cell divides, those tips get just a little bit shorter. And when they get too short, the cell stops dividing to protect itself from making mistakes.
But here's the thing. As important as telomeres are, they're just one piece of a much, much bigger puzzle. Modern science now sees aging not as one single thing, but as a whole collection of interconnected problems.
These are known as the hallmarks of aging. It includes the telomere shortening we just talked about, sure, but it also includes things like epigenetic alterations. That's basically changes in how your genes get read. There's also cellular senescence, the rise of so-called zombie cells, and even failures in our cellular power plants, the mitochondria. Now, we're going to zero in on two of these that are leading to some absolutely incredible breakthroughs: epigenetic alterations and cellular senescence. Okay, let's start with epigenetics. If we can't just count telomeres to get a biological age, then how in the world do scientists actually measure that cellular wear and tear? They use a tool that is just amazing. It's called an epigenetic clock. This is a super accurate test that looks at these little chemical tags on your DNA called methylation. The easiest way to think of them is like little post-it notes that tell your genes whether to turn on or turn off. And as we get older, the patterns of these little notes change in very predictable ways.
So how do you even build one of these things? Well, scientists take DNA from thousands and thousands of people. Then, using machine learning, they hunt for the specific spots on the genome where these methylation patterns reliably change with age. Then they bundle all that into a statistical model that can look at your DNA methylation and calculate a highly accurate biological age. It's pretty cool.
And this is where the power of these clocks really shines. If your epigenetic clock says you're older than your birthday suggests, that's a condition called age acceleration. And it's a huge warning sign. It's strongly linked to a higher risk for a whole range of chronic diseases. So this tool doesn't just tell you a number. It can help predict your future health. Which, of course, leads to the big question. If we can read the clock this well, can we do something about it? Can we actually step in and change the course of aging? And that brings us to our second hallmark: cellular senescence. So remember those cells that hit their Hayflick limit and stop dividing? That's senescence. But these cells don't just quietly retire. No, they become what scientists literally call zombie cells. They stop dividing, but they refuse to die. Instead, they just hang around, pumping out a toxic mix of inflammatory signals that damage the healthy tissue around them.
And you can see here just how dangerous they are. A single one of these zombie cells doesn't just sit there quietly. It secretes all these inflammatory compounds that can actually corrupt its healthy neighbors, turning them into zombie cells too. It's a chain reaction that just contributes to the breakdown of our tissues as we age. But what if you could perform a targeted zombie apocalypse at the cellular level? Well, that's the whole idea behind a revolutionary new class of drugs called senolytics. Their whole job is to seek and destroy these harmful senescent cells while leaving all the healthy cells perfectly fine.
And this isn't science fiction. Some of these are already in human trials. For instance, a combination of a cancer drug called dasatinib and quercetin, a natural compound you find in onions and apples, has shown some really promising results. Another one is ficitin, which is a flavonoid found in high amounts in strawberries, of all things, and it helps push these zombie cells to self-destruct. So let's step back for a second. What does all of this actually mean? How are these breakthroughs really creating a completely new vision for the future of aging? I think this quote really says it all. It captures the massive paradigm shift we're living through. These tools, like epigenetic clocks, are giving us a window not just to watch aging happen, but to finally understand its deep mechanisms in a way that suggests, yeah, we might one day be able to control it. And that really is the heart of it. For all of human history, aging was just seen as this inevitable decline, a fact of life. But this new science is reframing it entirely. It's a biological process. And like other biological processes, it might just be something we can influence, something we can manage, and maybe even something we can treat.
Which leaves us with a pretty fascinating final thought to chew on. The goal here isn't just about extending lifespan. It's about extending health span, the number of healthy, vibrant years we get to live. So as we get better and better at reading and maybe even editing our own cellular clocks, you have to wonder, will we one day have the power to choose our own health span? It's a wild thing to think about. Thanks so much for joining me.